Welcome back. So today we are going to be doing something slightly different, what we've done in the last few weeks. So instead of looking at a tutorial, I am presenting as if I was presenting at a scientific conference. In fact, these are slides that I've presented at a conference, and I'm adapting them to YouTube. And most of that adaptation is actually, I'm going to be breaking these things down into smaller chunks. I think a full, 42-minute presentation might not fly so well on YouTube, but I think if I break it down into multiple, 10 to 15-minute ones, I think this will make people happier and also having individual topics rather than this broad everything together. Okay, so today we're talking about the global microbiome, and so this was based on a paper that came out about a year ago, where we took data, and here's this map, shows you where the data comes from. And if you count, there's 14 different colors here, and many of them are human associated, but we also have other animal, mouse, cat, dog, and pig cuts, as well as environmental samples. And we took these, and the first thing we did is, and we used NGLS for this, so you can check that tutorial. We used NGLS to assemble, or we used MegaHit through NGLS to assemble, and then we predicted open reading frames, so potential genes in these contigs, and we ended up with about 2 billion of these ORFs, and we added the data from Progenomes 2. The meanwhile, Progenomes 3 is out, so in the meanwhile, actually the number of genomes has gone up by more than a factor of 10, but at the time we had Progenomes 2. And so we had 2.3 billion open reading frames, which we first dereplicated. So just when we had the exact same sequence from these two samples, we kept only one. And then we were down to 1 billion of these sequences. And then we further clustered them into multiple levels. The two most important ones was first this 95% nucleotide clustering, which corresponds to species level. And we're going to be calling these unigenes. And then at the other end, we had these protein families. And for us, a family means anything that's statistically significant with respect to shared ancestry. So this means that these sequences are homologous to each other. There's some shared common ancestor, but it might have been a long, long time ago. So why 95% for unigenes? And the reason is this is a proxy for species. So here we have in blue, so how close is the closest match if we look within a species versus if we look within a genus, but not the same species. And as you can see, there's an overlap between the blue and the orange curves, but 95%, which is was a standard threshold, actually corresponds pretty well to where these two lines intersect. So there's no perfect threshold, but if you're going to pick one, 95 is a great rule of thumb. Okay, so this together, we've called this the Global Microbial Gene Catalog, which we obviously often abbreviate to GMGC, and this is GMGC version 1. So often we will refer to this, and you'll see this in other videos as GMGC V1. And we have these 300 million unigenes, 30 million protein families, and as we talked in another video, we also have these metagenome assembled genomes, these MAGs that you can get and use for your own analysis. But now for the rest of my presentation today, I'm going to be talking about the analysis that we also did on our own data. And so the first question we asked is, where do these genes come from? And actually, this is the motivating question for a lot of this paper is, where do all these sequences come from? And so the first thing we saw is that here in these plots, so the width of these bars represents how much sharing is happening between the left and the right. And as you can see, there's really not that much. So these ribbons, they tend to be quite short. Most of the genes tend to be specific to their habitat, except when we zoom in within mammalian guts. And here I'm just going to zoom in on this right side of the plot, where you can see that now there is a lot more sharing. And in particular, a few years back, I think it's 2018, we had published this manuscript showing that in many metrics, the dog gut microbiome is the most similar one to the human one, compared to other mammals that we had analyzed at the time. However, if we now reanalyze this, having more data, we actually see that the cat is also a great match to humans. And actually, if you just count, you can see that there's actually slightly more sharing between the cat and the human. But in any way, it seems that it's these pets, the cats and dogs, rather than pigs or mice, whose microbiome really resembles ours the most. So this was already pretty interesting. But then the other thing we observed is that most genes are rare. And so here what we're showing is, so the number of genes that we only detect one single time, 2 times, 3 times, 4 times, etc. And as you can see, it goes down quite quickly. So the median number is 10. So most genes are detected 10 or fewer times out of over 10,000 samples. And so this is actually true whether we're speaking about unigenes or whether we're speaking about gene families. So then the exact numbers, are different. Obviously, we have 100 times more unigenes than families, but the pattern stays the same. And actually, in the manuscript, we show that even other intermediate thresholds, the pattern stays the same. That this number goes down very quickly. It also follows these, fairly straight lines with the slope being very close to -1. And it is true that this is a log-log plot, and everything looks linear on a log-log plot, but these models also really do look exactly how you'd expect if you assume that most genes are following neutral evolution, in the sense that if you replace one variant by another, it doesn't really matter for selection. The other thing that becomes apparent is that we do have these genes that we see only 1 time and 2 times. And every time we sequence a new sample, even in the human gut metagenome, and here it's the most widely explored model system for microbiome research, because we're very species centric. We've probably saturated in terms of things that are seen that are common. So genes that are seen in majority of at least 5% or 1% of samples, we've seen them all more or less. But still, despite having sequenced thousands of samples, every sample has new genes, and it might actually be the case that if every single individual, so every one of you that's watching this video, would have that your own specific genes in your microbiome, and there's been several studies that have shown actually that even over relatively short periods of time, and certainly during human lifetime, your microbiome will have evolved new genes. And again, maybe the majority of them are not doing anything special. This is just neutral evolution, but this diversity of genes is present, and if maybe every once in a while, it's possible that some of this will start getting selected. But we had these inklings of a lot of this matches this model where there's not a lot of selection. So can we look at this a bit more closely, and can we pick out other metrics of selection? And so here we looked at a couple of different ones. So for example, how much is operon structure selected for, or conserved? So if we see, and this just means often genes are next in microbial genomes, in bacterial genomes, are next to other genes that are functionally related. So how much selection is there for this? And here we've grouped them into the genes into categories. So these rare genes, fewer than 10 detections, genes that are pretty still kind of rare but not so much, and then the very frequent genes. And as you can see, there's more conservation, so more selection as you go to these more common genes. Here's another metric is looking now at single nucleotide variation within a gene family. So as you said, we have these families, so we can see what's happening in these families, and we can see that this is happening again. Measures of positive selection, they go up for more common versus less common genes, and here we also took the E. coli pangenome, because we actually have a lot of E. coli genomes, and there's a couple of reasons why we took this one, but the main one is just it's actually the one where there's more genomes, and so we couldn't do this analysis. We didn't have enough numbers for any other species, but the pattern follows more or less the same. So, and then even if we look now only within strains, we get again the same. So different metrics of what does it mean for a gene to be selected. It seems that we do have, these rare genes. They seem to be evolving neutrally, and maybe there's another class of genes that are very prevalent where selection is acting in a different way. So still asking the question, so where do these genes come from? And this we also looked at how do these families group the genes. And here and you can see this is a log plot. So even in a log plot, it goes down very quickly. And so we have a lot of singleton families, but then it goes up so that the very largest families are really much, much larger. And so that we only need 0.6% of families, and they already contained about half of the unigenes. So half of these are really come from diversification within a relatively small number of families. We're talking about 0.5%, but of 30 million. So we're still talking about hundreds of thousands. But it's mostly diversification within existing families rather than a lot of different functions and a lot of different completely novel structures. So in this spirit, we looked at this relationship between function and between diversity of function, using here the families as a proxy for functions, and then in the species level unigenes. And here, I'm going to be fully honest. I mean, I think there's still, this is, for me, this is interesting, but I still don't really understand exactly what's going on here. And I think there's probably going to be some follow-up work from us or from, others trying to explain and trying to dig into this. But here's what I'm showing you is, so here, if we look at a particular sample, how many unigenes do we have per species? So how big is the local pangenome of that on average in a sample? So in a microbiome, and as well as how many different species level unigenes do we have per gene family? So this measures, if we think of the families are a proxy for function, this measures how much redundancy we have. So if there was no redundancy, within a community, then this would be 1, right? We have a single unigene in that family in the whole community. And if we have more unigenes that are related to each other, even if they're in different species, there's probably a little bit more redundancy there. And so what's interesting here is that for most of these, we see these peaks, that are fairly well defined. And actually, we can use just these richest numbers can be used to distinguish and to classify the different habitats with impressive accuracy of above 80%. And it's interesting too that if we look at, for example, here the marine seems to follow a bimodal distribution, and the soil, you can also see it seems to be a conceptual case where we don't have a defined peak, but we have a couple of different peaks. And actually, that's a little bit of an illusion, because what's happening here is that if we look at the marine samples, what we see is that if we have the surface samples, as well as the CM, they form this first peak, and then this mesopelagic zone, which is where it's deep enough where there's not a lot of light, it forms the second peak. So it looked bimodal because indeed it was a mixture model of these two. And then I'm actually not so familiar with soil, but it also this very broad distribution actually can be partitioned into different kinds of soils. So there's these signatures that even very different samples from across the world, they seem to this pattern of how many conspecific genes, so the number of unique genes per species or the number of unique genes per family seems to be a property of the environment, and we see this, we report this, it's very robust, but we don't really understand the mechanism at this point. So we also, as I said, this is also a resource. You can go to the website. We'll probably do a video about how, using the website. So here, just showing you that we do have a website. You can, put in whatever your query is. It will search it, sort of a variant on BLAST, and you get these results. All of the data is available for downloading, and you can, if for more advanced users, or if you know what this means, we have an API. You can use, it's a REST API. You can use, for access programmatically. So everything's there. So we've done some work. We've made data available for download for everybody else. Watch the other tutorial if you want to see how to get to the mags. But just to summarize, we have these 300 million unique genes, which we cluster into 30 million families. Most of them are rare, and most of the variation is within these large families, and it's mostly likely neutral or neutral. So we have this as a resource for others. And here today I've managed to just show you a few of the highlights from the manuscript. So we do have a paper with a lot more results. I do want to thank a lot of the other people, especially Jaime and Per Bork, also the funders. And I want to thank you. And if you have any questions, please ask. We can email, you can send a DM, you can comment below, we can do a Zoom call. I'm happy to hear from everyone, and I hope to see you soon.